All right. Thank you for attending the HIV Grand Rounds, which is put together by the UCLA AIDS Institute and the Center for HIV Identification, Prevention, and Treatment Services. My name is Oluwadamilola Jolayemi, and I am going to be here waiting and making sure everybody comes in time. We'll be starting in about 2 minutes with our introduction, with our speaker introduction. Our title of our presentation today is The Enemy of My Enemy, which is bacteriophage therapy to treat multi-drug resistant bacterial infections. And we have our 2 amazing speakers today, and we'll be hearing from in a little bit. So we're waiting for everybody to trickle in. Once we have a good number of people, we will get started. Thank you for your patience. Good morning. Thank you for attending the HIV Grand Rounds. This is put together by CHIPS, UCLA CHIPTS, and the AIDS Institute. So we're going to go ahead and get started in a little bit. But before we get started, I wanted to go over some housekeeping items. My name is Damilola Jolayemi, and I'm here with UCLA Center for HIV Identification, Prevention, and Treatment Services. And so just before we get started, just so you know, the presentation would take about 45 minutes. And during that time, please feel free to use the chat to ask questions or to post comments for the speakers. All of the questions and comments will be addressed at the end of the lecture. So please be patient and wait for your questions to be addressed. If they're not addressed during the lecture due to short time, we will definitely follow up with you after the lecture. So please feel free to use the chat. If you want to send messages privately, feel free to do that as well and send it directly to the host, which is me. And then we're going to share that with the speakers. So thank you again for attending. We're going to go ahead and get started. I'm going to introduce Dr. Steve Shapta, who is the director of CHIPS, who will go ahead and introduce the speakers. Dr. Shapta, go ahead and take it away.
Thanks, Dami. It's great to be here today. It's especially wonderful to have our distinguished speakers today. I do want to thank you both for joining us here at UCLA HIV Grand Rounds, and I do want to recognize that not only is CHIPS sponsoring this, but this is also an event sponsored by the UCLA AIDS Institute. So we're very grateful to both centers for the support. Today, the enemy of my enemy, bacteriophage therapy to treat multi-drug resistant behavioral infections. Our speakers are Steffanie Strathdee and Chip Schooley. Steffanie is the Associate Dean of Global Health Sciences and the Harold Simon Distinguished Professor in the Department of Medicine at UCSD. She co-directs she has 3 centers. She co-directs UCSD's new Center for Innovative Phage Application and Therapeutics, IPATH, which is this nice graphic at the bottom here on the right, the Global Health Institute, and the International Core of UCSD's Center for AIDS Research. Unbelievably, Dr. Strathdee has over 600 peer-reviewed papers and recently has moved from working in HIV prevention and addiction work to move bacteriophage therapy into clinical trials at IPATH. She co-mentored her memoir, The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug, which was published in 2019. She, together with Chip Schooley, who will be speaking with us on this topic, Chip is the interim faculty director of global education, the senior director of international initiatives, professor of medicine in the Division of Infectious Diseases and Global Public Health, and co-director of the Center for Innovative Phage Applications and Therapeutics with Dr. Strathdee. Dr. Schooley is a graduate of Hopkins University School of Medicine. He completed his residency at Hopkins and a fellowship and fellowships at NIAID and Mass General Hospital. He joined the faculty of Harvard Medical School in '81 and shifted his research focus from herpes group viruses as recognition of the AIDS epidemic developed. He left Harvard to go to be the division director at the University of Colorado in infectious diseases. He was elected chair of NIH's AIDS Clinical Trials Group in '95 and led that group until 2002, expanding the ACTG from domestic research primarily to one with a global reach. In 2005, he joined the faculty at the University of California, San Diego, where he served as the head of the Division of Infectious Diseases until 2017. He's editor-in-chief of Clinical Infectious Diseases. I don't know where he finds time to do this. These two people are really unique. And his research interests are in the diagnosis, pathogenesis, and therapy of viral infections and global health. So with that, I think I'll turn the talk over to Steffanie, Dr. Strathdee.
Great. Thank you very much, Steve. It's just a real pleasure for Chip and I to have this opportunity to present to you today. Seeing lots of familiar faces and names in the panel, so I'm looking forward to our discussion later on. This is an unusual presentation. I'm going to be talking about a field that's been relatively new for me. I'm going to first have a couple of disclosures. My husband and I now hold stock in a phage company. I'm not going to be talking about that today. Any of the patient photos shown are used with permission. Most of them are my husband, because actually this is a personal story that led to the expansion of a field that is still emerging. And it began for me and my family in Egypt when my husband, Tom Patterson, who many of you who are AIDS researchers will know as the founding editor of the journal AIDS and Behavior. We go on conferences together, then we take a couple personal days, and here we are in Egypt. This is November of 2015. We thought that we'd go and see the Valley of the Kings, and Tom looked healthy. I didn't think that there was anything wrong. He was certainly overweight, but after this wonderful meal on top of a cruise ship, he developed intense abdominal pain and vomiting, and it didn't get any better. I actually called Chip Schooley, who my co-presenter this morning, as one of the first things I did, and asked him for advice. And he was asking about his symptoms, and he said, well, it sounds like he needs medical attention. It's been over 24 hours. He's losing fluids. He's going to need fluids for sure. Get him to the closest hospital and go to where the expats go. Well, there was no hospital in Luxor where we were based. But luckily they did have a CT clinic, and the next day they rolled him in there, and you can see he's in terrible pain. And what they had diagnosed at this time was pancreatitis, which is essentially an inflammation of the pancreas. And I thought, okay, that's good. They can fix that, right? But then Chip explained to me that that's just a symptom of a much larger problem. And in fact, whatever they saw on this CT scan, they didn't like. And immediately they began to be more helpful in terms of medevacing him first to Germany and then back home to San Diego. In Germany, you can see we're in the ICU and we're in full PPE. Everybody knows what PPE is now because we're facing yet another pandemic. And Tom was in and out of consciousness, and I started to get worried for the first time. Here is where they did an endoscopy and found that a gallstone had lodged itself within his common bile duct and caused an abscess to form the size of a small football in his abdomen. That wasn't all. The doctor approached me and said, look, he approached me with this putrid fluid inside a flask. And he said, this abscess has been there for quite a while. If it had just formed, this fluid would be clear, but it's not. It's putrid. There's something growing in it. And we better hope that it's a garden variety microorganism, because you've just come from Egypt, which is why we have you in isolation, because we're hoping that it's not multi-drug resistant. Well, bad news. A couple days later, the culture comes back and it's Acinetobacter baumannii. And unfortunately, that's known as one of the most deadly bacterial infections. It's very prone to multi-drug resistance. And this is an organism I used to plate in my Petri dishes back in the 1980s when I was doing my undergraduate degree in microbiology at the University of Toronto. And all we needed then was a lab coat and gloves. But over the last few decades, it's gotten to be quite a wily pathogen because it's got sticky little fingers. It's got a biofilm formation that it's prone to, and it can even stick to body lice and hospital linens. So when physicians are facing this organism, they're often providing the patient with some of the most heavy duty antibiotics. In fact, The antibiogram, which is essentially the antibiotic susceptibility profile, was run by the German doctors, and it came back with multiple resistance. So there was only partial sensitivity to minocycline and colistin, and all of those are need to be infused into the patient. So it was bad news from the beginning. It was a highly resistant organism. And so they immediately put him on these antibiotics, stabilized him, and then got him back home to San Diego. So by this time, I started to realize that my husband was actually this poster child for this post-antibiotic era that we're entering, that the WHO and the CDC have been warning us about for years. It's estimated by 2050, 10 million people per year, that's one person every 3 seconds, is going to be dying from a multi-drug resistant bacterial infection, and that will cost $100 trillion to society through the loss of productivity. Now, how did we get here? Well, I won't get into this in a lot of detail. We can talk about this in the discussion, but it isn't that most antibiotics are being misused in medicine. About 70% of antibiotics being used in the US are used in agribusiness, and it's really to make animals grow fatter faster. This was discovered after antibiotics were developed, that especially in poultry early on, that animals that were given antibiotics fattened up really quickly. Now the US and Canada and some other countries have made it more difficult to use antibiotics as a growth promoter, but they are still used off-label in this way. And so that's one of the biggest sources of antibiotic resistance, because when you're using the same antibiotics in animals that you're using to treat human bacterial infections, resistance is prone to occur. So back to our story. Well, by the time Tom got medevaced back home to UC San Diego, where that I'm faculty and my colleagues like Dr. Schooley were treating him, he was really sick. He was in and out of a coma at various points, and he was considered to be too sick for surgery and too weak for surgery. This organism was spreading. So what they decided to do was to enlist the help of interventional radiologists to essentially, poke holes in his abdomen, put catheters in there to try to siphon off that infected fluid, with the hopes that this abscess would shrink and his immune system would kick in. Unfortunately, these catheters are prone to clogging with all of the debris that is, oozing out of them. And the organism continued to spread and form more fluid collections in his abdomen. So by the time this photo was taken, he had 5 different catheters in his abdomen, as well as a feeding tube. And you can see that he's on a ventilator, and he was on 3 different medications or pressors to keep his heart pumping. In essence, he was dying. And this is because one of those drains slipped inside him one day and poured all of that infected fluid into his bloodstream and into his abdomen, and he immediately went into septic shock right before my eyes. So this photo was actually taken after my colleagues told us that he wasn't going to make it, and that he was going to be, slipping away a little by little. And I hadn't given up yet. I had a conversation with him, and this photo was snapped by my stepdaughter, Tom's oldest daughter. And I said, honey, I know you're doing everything that you can and you're really tired, but if you want to live, I need you to fight with everything that you've got, but also try to squeeze my hand and let me know if you want to live, because I'll try to see if I can find something, anything that will fight this thing. So even though he was in a coma, and believe it or not, he was having a hallucination and he thought he was a snake, I learned later on, he said it took him a while to figure out how to wrap his tail around my arm and squeeze it so that he could tell me that he wanted to live. But he eventually squeezed my hand really hard. I got really excited. And then I thought, oh crap, we have some of the best infectious disease physicians in our university hospital. How the heck am I going to do something here? But the good news is I do know how to do research, and I went home and I started doing research by just plugging in keywords into PubMed. And around this time, Maria Ekstrand, who's part of the UCSF CFAR, who's a colleague of mine, sent me a message saying, you know, I found something that a friend of mine told me, that she had a multi-drug resistant staph infection and went to the former Soviet Union to get something called phage therapy, and it worked. This sounds a little crazy, but maybe it would work to help Tom. Well, I had just stumbled across this paper, Emerging Therapies for Multi-Drug Resistant Acinetobacter baumannii. And I kind of thought, wow, you know, this is a little bit odd. Maybe I should look into this. But then quickly I realized that phages, which are viruses that have naturally evolved to attack bacteria, were not approved for treatment of bacterial infections in the Western world, including the US. And so I wrote Dr. Schooley an email and said, you know, what do you think about phage therapy to treat Tom? And he said, well, what an interesting and intriguing idea. If you can find some phages that would be a match to Tom's bacterial isolate, I can contact the FDA and see if they'll give us permission for compassionate use. So that's how our story started. But I have to give you a little bit more background before we get into the clinical and infectious disease realm here. Interestingly, phages or bacteriophages were discovered over 100 years ago. They have a very interesting history. Researchers like Carl Hankin on the top left and another Russian researcher on the right had seen activity or bacteriolytic activity well, you know, before the real discoverer Felix de Herel. What they did was they passed organisms and, you know, and they took feces from people who had survived cholera and put it through a Pasteur filter. And that filtrate they saw was able to kill bacteria in a Petri dish or in a bacterial suspension. And it wasn't until Felix de Herel, a self-taught French-Canadian microbiologist in 1917, did that same experiment, but then he deduced that whatever it was must be smaller than a bacterium because it can go through a Pasteur filter. Therefore, it must be a parasite of a bacterium because it can kill these bacteria, and therefore it must be a virus. So he called these organisms bacteriophage. Of course, only the light microscope was available at this time, and so you couldn't see these phages are 100 times smaller than bacteria. But he was attacked for this view. Other people thought that maybe this bacteria eating effect was caused by an enzyme. And it wasn't until much later that he was vindicated. But Felix went on to use phage preparations to treat bacterial infections in humans and in animals and had some success. He was actually the inspiration for the Pulitzer Prize winning novel in 1925. And it was, you know, something of a heyday at this point. But then penicillin came on the scene. It was ushered into the mainstream because of World War II. And phages in the West were kind of relegated to the back burner. Now, they continued to be used in the former Soviet Union and in Poland, where they're still used to this day. And in fact, that really cast a pall over the field of phage therapy for a time because it was dubbed as Soviet science, Soviet medicine, and of course, the Soviets were the enemy. And so that was another nail in the coffin for phage therapy as to why it was forgotten in the West. So it wasn't until the electron microscope was developed in the early 1940s in Germany that phages could be visualized for the first time. This is a scanning electron microscope that was done in more recent years. The bacterium is stained in orange, the phages are stained in green, and these are colophages that are the kind that people are most familiar with, but they come in all shapes and sizes we know now. And you see they look a little bit like alien spiders. They attach to a bacterium through a receptor, they drill into it, their genetic material, usually DNA, enters the bacterial cell, takes it over, turns it into a phage manufacturing plant. And if it's the lytic cycle, when given the kill signal, all of the baby phages or virions burst out of the bacterium and go on and attack new bacteria that they are a match to. Now, they only attack the bacteria that have their receptors, so they don't actually cause harm to the microbiome, because bacteria that don't have a receptor are untouched. So essentially, we know now that about 30 billion phages move in and out of our body every single day. It's just a matter of finding the right ones to kill the bacteria that are causing harm. So here is exactly how you source phage. Here is a flask of sewage. You find phage wherever you find bacteria. They're an estimated 10 to the power of 31 or 10 million trillion phages on the planet. They're found everywhere. They're on our skin, they're in soil, they're in water. But if you have a gut infection and you want to, kill that organism that's causing the dysbiosis or whatever, you can source phage easily from sewage, because there's a lot of bacteria in sewage. So you get a drop of sewage and you put a drop of it on your Petri dish. And this is our pal Acinetobacter baumannii. You can see all those glossy colonies. You incubate it for 24 to 48 hours. And if it comes back looking like Swiss cheese, you get excited, because even though you can't see the phage with the naked eye, you can see that they've been gobbling up those bacterial colonies. And this is evidence that the phage are a match. So you pluck out those phage with a pipette. This is the old-fashioned plaque assay that I'm describing, that it can be done by high school students. And then you add more bacterial suspension and grow up those phage, and they can be used to treat bacterial infections. So I thought, wow, the more I read about this, the more excited I got. I knew what phages were from my rusty old degree in microbiology, but I never knew that they had been used to treat bacterial infections. So I thought, okay, CHIPS on board. The hospital says if we can get the FDA approval, once we find phages that match, we can use this to treat Tom, right? Well, it was more tricky than that, right? Because with 10 million trillion phages on the planet, how are you going to find the perfect predator to kill them? Well, I went back to the internet and I made a list of phage researchers that were studying Acinetobacter baumannii and phage. And I made my list only to the US, because I knew we didn't have much time, because this is what Tom looked like. This t-shirt strewn across him was given to us by one of our postdocs. I survived Iraqibacter, it says. Iraqibacter is the nickname for Acinetobacter baumannii, since so many veterans came back from the Middle East with this infection in the early 2000s. And I thought, well, maybe I can, appeal to the heartstrings of some of these researchers. I wrote them cold, and within 24 hours, I heard back from Dr. Ryland Young from Texas A&M University, who said, I've been studying phage my whole life. It would be great if phage therapy came back and made, a mark in the West, and maybe this is the case that can do it. So do you have his bacterial isolate? If you do, send it to me and I'll turn my lab into a command center. So the people pictured on the right-hand side worked in Rye's lab. The woman with the necklace was a PhD student at the time. She slept in the laboratory, and lo and behold, they went through barnyard waste and, samples from duck ponds and found 4 phages that matched. Now, when I told Chip this, he was really elated, and he put some resources together from the literature and got ready to make a case to the FDA to say, could we use these phages found by Texas A&M to treat Tom? She contacted Dr. Cara Fiore, who at that point was knee deep in fecal transplant requests at the FDA and was looking for a change. And he expected to have to tell her all about what phage therapy was and why we should give this a try in Tom's case. And she actually knew all about this literature. She said, we've been waiting for a case like this for a patient who was dying, who was out of antibiotic options, a family that's willing to give a try, a physician like you who's willing to cut through the red tape, and a university hospital and a phage research community that are willing to find phage in time. So she said, yeah, we will give you compassionate use, but you have to get some information for us first. So in the meantime, she told us that there was actually the Army and the Navy were working on phage and had phage libraries, and maybe they would help. So Chip approached This is Lieutenant Commander Theron Hamilton, who has an MD, PhD, who at the time was overseeing the U.S. Navy Biological Defense Research Directorate. And he was able to convince Theron that, you know, he should look to see if any of the phages that they had in their library that had been sourced from the bilges of ships around the world could be a match for Tom. And he enlisted the help of Dr. Biswajit Biswas on the right-hand side, who is affectionately known as the phage whisperer, I kid you not. And Biswajit actually found 4 phages that matched. Now, at this point in our story, you might be wondering, well, you already had the phages from Texas A&M. Why do you need more phages? Well, the theory goes that, you know, if you only have one phage or phages that are only hitting the same receptor, then the bacteria that's being attacked by the phage could become resistant very quickly. So we wanted to have multiple phages in a phage cocktail. We didn't have the time to sequence them to know whether they were hitting different receptors or not. But we thought at this point, the more phages, the merrier. Well, there was another wrinkle, because it turned out that when the FDA asked us to determine how much endotoxin was in the phage preparation, it was very high. Now, endotoxin is essentially the lipopolysaccharide layer of the gram-negative bacterial cell that is elicited when the phage and the bacteria are attacking one another. And so when you're making and growing up a lot of phage, this is the bacterial debris from the bacteria that are lysed. And so it can actually be toxic to the patient and could elicit septic shock. So we enlisted the help of San Diego State University, who just like fortuitously had a system to remove endotoxin with the organism that we were dealing with, and they were able to do this over the weekend. So we had this phage preparation now, but Dr. Schooley in the meantime was really grappling with some other questions, and these are still questions that we're dealing with to this day. What is the dose when the drug is alive? I mean, we know how much phage you're administering to the patient, but those phage are going to multiply as long as they have a bacterial target. So you really are not sure how much the patient is actually getting. What routes of administration are best? Chip was convinced that we needed to treat Tom intravenously, because by this point he was fully colonized with this multi-drug resistant bacterial infection that by now was pan-resistant to all of the antibiotics that we had at our disposal. And then how often, how long should we treat the patient? Chip consulted with Dr. Maya Mirabishvili, who was trained at the Eliava Foundation. And that is actually the first phage therapy center to be formed in the world in what is now Tbilisi, Georgia. And she gave him some advice on dosing. He also consulted Dr. Carl Merrill, who used to work for the intramural program at the NIH and was a phage researcher, who also said, no, you really need to go intravenous, because if there's a hidden reservoir of bacteria that the phage don't reach, that it could become resistant right away, and then it would be game over. Well, here is the phage preparation that our investigational pharmacy prepared. It took them all day once they got the phage that was purified by San Diego State researchers. I was pacing the floor knowing that Tom was running out of time. I actually signed the consent form for kidney dialysis the day that we started phage therapy. But, you know, the investigational pharmacists, this is the first time that they dealt with a drug that was alive. They're used to preparing, antiretrovirals for, HIV clinical trials and the like. This was a very unique situation for them. And this is what Tom looked like the day we started phage therapy. This is March 15th of 2016. So he'd been in the hospital 4 months. And as you can see, he was in a deep coma and he was not being roused at this point. He was entering multi-drug and multi-system organ failure, and we were very scared. Now, this is Chip and our colleague, Dr. Randy Taplitz. They're smiling for the camera because they knew that essentially they were making medical history. Tom was the first person that we know of in the US to be treated intravenously with a phage cocktail to treat a multi-drug resistant bacterial infection. And we decided that we would collect specimens before and after, so that if Tom passed, that at least he would be moving the field forward. as you can imagine, it was the scariest day of my life. We had first the interventional radiologist, Dr. Pacell, take the Texas phages that were ready first and insert those into the catheters in his abdomen. And this was 10 to the 9 PFU per mil, or 1 billion phages per dose every 2 hours. And Chip said, if he survives this, then we'll get the Navy phages, which were more virulent, and we will inject those into his PICC line. And that was done a couple of days later because he was holding on. And then, believe it or not, 3 days later, Tom lifted his head off the pillow and kissed his daughter's hand. Nobody really knew what was going to happen. And in fact, one of my colleagues in the ICU said to us, you know, this is like the last minute of the football game where like, you're underwater and you don't know if you're going to be able to make that last touchdown, and you're throwing the ball 100 yards down the field and the quarterback is blindfolded and somebody catches the ball. So it was a really tremendous moment, very emotional. Tom had a very rocky recovery because he was lost 100 pounds. He was so deconditioned, but he left the hospital 9 months after this ordeal began in August of 2016. It was a tremendous day. He's wearing his Superman t-shirt. And a couple months later, he was happy to meet Lieutenant Commander Theron Hamilton, who oversaw the preparation of the Navy phages. And in the bottom left, you'll see that Tom's case report was published by Dr. Schooley and colleagues in AAC in 2017. And as a side note, you can ask Chip in the Q&A about the ordeal we had about trying to find the right home for this paper. That's quite an interesting story. And here is actually another electron micrograph. This is a scanning electron micrograph that was prepared by the Department of Homeland Security at the request of the Navy. This is Tom's Acinetobacter baumannii isolate stained in blue being attacked by the Navy phages stained in green. So it's a pretty amazing micrograph. This is blown up 100,000 times. So after Tom's case was presented at the 100th anniversary of the discovery of bacteriophages in, I guess that was 2017 of April, the story went viral, but in a good way. It was picked up by the press all over the world, and it was also covered in a number of medical journals like JAMA and Lancet. And I started getting phone calls, as did Chip, from patients all around the world, their families, and sometimes their doctors. People wanted phage therapy. People had a superbug infection. Could they get phage therapy too? Well, of course, total strangers had stepped up around the world to help my husband. We didn't need all of the phages that were offered, but we had phages that were offered to us from researchers in India, Switzerland, Belgium. It was quite incredible. And I thought, well, you know, maybe we can try to help them too. And Chip agreed. And each time we had to source phages for patients that were running out of options. And in some cases, we were able to help. And in some cases, we weren't. We helped enough patients, though, that by 2018, the chancellor at UC San Diego, Pradeep Khosla, gave us seed funding to start what is now the first dedicated phage therapy institute in North America, IPATH, the Center for Innovative Phage Applications and Therapeutics. And I co-direct this along with Chip and Drs. Saima Aslam, Dr. Connie Benson, and Dr. David Pride are key staff, but all of the infectious disease physicians at UC San Diego Health are aware of phage therapy and often refer patients to us. The day that we opened, Science Magazine published a commentary saying that this was a game changer in the field. Since this time, we treated many other infections. This is actually just a partial list because I wanted you to see that we're not just treating patients with Acinetobacter baumannii. Tom was patient number 1, but we treated patients with Pseudomonas aeruginosa, Staph aureus, ESBL E. coli, and others. And Dr. Benson, who is our Mycobacterium expert, has been involved in the treatment of over 25 cases of Mycobacterium abscessus. I wanted to also show you another interesting case. This is the Mayo Clinic's first experience with phage therapy. These pictures were sent to me by the patient, John Haverty, who'd had a prosthetic joint implanted in his knee, and he had slipped, and his incision opened up, and it got infected with a multi-drug resistant Klebsiella pneumonia bacterium. And he'd had 19 surgeries by January of 2019 to try to remove this infected tissue. They were unsuccessful. They told him he was going to have his leg amputated. The photo in the middle was taken right before that happened. And his wife happened to stumble across our story and approached a phage company who sourced phages that matched John's isolate. And a researcher who had actually worked in a phage lab, who was a transplant or actually an ortho ID expert, Dr. Gina Suh, had just started working at the Mayo Clinic, and said that she would give this a try. And after 2 infusions, that's the picture on the right-hand side. In fact, when Mr. Haverty said that he wanted to reach me, we contacted each other through social media. I gave him a call, and I got voicemail, and he called me back, and he says, "Sorry, I was out mowing the lawn." I mean, honestly, that's how quickly it worked. It's really quite remarkable. Now, of course, these are just case reports and sometimes case series. We've published a large number of them, and there have been others published around the world since. But we have to go along a lot further if we're going to advance this and bring it to scale. In fact, if you look at the phage referrals that have come into IPATH, you see since we opened in June of 2018, we've had over 1,100, that's on the left-hand side, 1,100 referrals. Now, not all of these are eligible for phage therapy because right now it's still considered an experimental treatment by the FDA because clinical trials have not yet been completed to show that it's efficacious. So of the cases where phage hunts were recommended, and 204 cases, sometimes we weren't able to get a bacterial isolate in time, or the patient, if they have prostatitis, for example, it's sometimes difficult to obtain a culture. A phage hunt was initiated in 170 cases. In many of these cases, phage were not found in time. The patient died, or in some cases, the infection resolved. But lytic phages were found in 82 cases. And so we've gone on to have some success, but some failures. So overall, 2.6% is of the requests that we get in are being treated. Now, obviously, that's very inefficient. So we need to improve upon this. And just again, to show you the kinds of requests we're getting in are now a myriad of bacterial infections, some of which are easy to source phages for, and some of them are more difficult. So what this really tells us, though, is that what we need to do is to build an ever-expanding phage library that maps onto a library of multi-drug resistant organisms. And in some cases, like Staph or Pseudomonas aeruginosa, it's maybe possible to have a fixed phage cocktail so that you can cover the majority of circulating isolates around the world. But in an organism like Acinetobacter baumannii, you actually have to match the phage to the specific isolate. So like Tom's Acinetobacter baumannii, not anybody else's, because it's got the phage have a very, very narrow host tropism. In the case of Mycobacterium abscessus, it's even more difficult because we can only seem to find temperate phages rather than lytic phages in the majority of those cases. And those I describe in our book as the sleepy kind of phages that integrate their DNA into the bacterial cell DNA and hit the snooze button. We don't want to use those phages because first, they don't kill the bacterial cell. Second, they often carry toxin genes or antimicrobial resistance genes. And third, they even can collude with the bacterial cell to make that bacterial cell resistant to attack from other phages. So we either want to exclude those phages and use only lytic phages, but if we can't find anything but the temperate phages, we need to be more creative. And in fact, I'll tell you about a case in a minute where genetically engineered phage was prepared to treat a patient. Chip and I co-authored a commentary in Nature Microbiology in May of 2020 about how we think that the path forward should occur in terms of moving phage into clinical trials and what kinds of translational research needs to be done to do that. And Chip is an expert on this area, so I'll wait for the Q&A so that he can tell you more about how he thinks the research needs to be conducted. We were really pleased, though, in December of 2019, right before COVID hit, the NIH and specifically NIAID funded their first phage therapy trial through the Antimicrobial Resistance Leadership Group, which, as many of you are aware, is a network of research groups around the US that have been primarily focused on new antibiotics and getting those through the pipeline. But since the pipeline for antibiotics is drawing up, they got excited about phage thanks to Chip's discussions with them. And this is an adaptive phase 2 trial. And enrollment is going to start in early 2022, and Chip is the PI, and Dr. Benson and Dr. Pride are playing key roles in this as well. So it's a multi-site trial, and it will start enrolling at IPATH in the months ahead. And we're very excited about it. It was been slowed down due to COVID. So in terms of the other kinds of studies that we need to do, we need to assess pharmacokinetics, pharmacodynamics, valency, dose, the best routes of administration. In Tom's case and in others, we've seen synergy with antibiotics and phage. And so trying to predict which phage go with which antibiotics and which phage go best with other kinds of phage. Those kinds of bioinformatics and machine learning approaches are definitely needed. We need lots of clinical trials to determine efficacy with different kinds of multi-drug resistant infections. We need a phage library, as I pointed out, to be able to source phage faster. And I want to end with a last case report that describes the first genetically modified phage cocktail to treat a patient with a disseminated drug-resistant Mycobacterium abscessus infection. This is Isabel, who I've come to know. She lives in the UK. She has cystic fibrosis. She'd had a double lung transplant, and her new lungs were being attacked by a multi-drug resistant Mycobacterium abscessus infection. And of course, many of you will be aware that this is a cousin to Mycobacterium tuberculosis. It's an intracellular pathogen, very difficult to treat. And she was in hospice. Isabel's mom heard about Tom's case, reached out to her physician, who reached across the pond, and my colleagues, Dr. Schooley and Dr. Benson, got involved, but they also collaborated with Dr. Graham Hatfull in his lab from the University of Pittsburgh. Graham has a program called the CPhages program. There are actually 4 CPhages programs in the UC system, one at UCLA and one at UCSD as well. This is essentially an undergraduate program where students are learning how to source phage, and all of the phage that they find are going into a phage library, and they had a big phage library at University of Pittsburgh as a result that were crowdsourced by students, and these were all Mycobacterium phages. They were able to find some phages that were a match for Isabel's bacterial isolate, but they only found one lytic phage. It was sourced from a rotting eggplant in South Africa by a student there. The other 2 were temperate phages, and because we wanted to use a phage cocktail for the reasons I described earlier, Dr. Hatfull and colleagues clipped out the repressor gene of those temperate phages, forcing them to become lytic. And now we had the first genetically modified phage cocktail. Luckily, the UK government was convinced by Dr. Schooley and colleagues that this was not, going to be a genetically modified organism, because they actually took away a gene rather than adding a gene, and they allowed this to go forward. And Isabel actually left the hospital within a week. This is how quickly she responded. She's had a difficult course of infection as well, but it does lend hope that maybe someday Mycobacterium tuberculosis could be treated with phage. And in fact, Dr. Benson is working on that with Dr. Hatfull. And also it's opened the door for biotechs and pharmas that get very excited because it's much easier to patent genetically modified or synthetic phage. So with that, I'm going to wrap up, but I want you all to be aware that this pandemic of multi-drug resistant organisms is already here. It's worsened under the COVID pandemic. Just a few weeks ago, there was a paper published that showed that there was a significant increase in a number of hospital-acquired infections as a result of COVID. And so it's going to be a problem that we're facing. And HIV-infected patients are going to be especially at risk as well. So with that, I'll stop there and allow my colleague, Dr. Schooley, to join in. And I know that some of my other IPATH colleagues have joined as well. It's just a pleasure to be able to present this to you today. And I want to also thank the global village of researchers and physicians and nurses who really stepped up the plate to save my husband's life. And if we can do this for one man, we can do it for the planet. My husband and I realized how privileged that we were to receive the help that we did, and we decided to write our story as a memoir. It's done very well. It looks like it might become a Hollywood movie. I can't talk much more about that, but it's like turning the worst thing that could ever happen to you into something that maybe is helping other people. So with that, I'll stop and see what questions you might have for us. Thank you.
Okay, Steph, you want to like stop your share?
Yep.
Thank you. Thank you. That's awesome. Sam Griffith is here. He asked, how will we let me say this. Big reach you guys have. How will we prevent what's happened with antibiotics to happen with phage in terms of resistance? And I actually I'd like to tack on to that. Can you describe the process of how a bacteria becomes resistant to a phage?
Chip, where are you? I know I could make a stab, but you could do this a lot better than me.
Sure. No, bacteria and phages have been dancing with each other for about 300 million years, and the natural history of this interaction is that when a bacterial population is attacked by a phage population, the bacteria evolve around those phages by either modifying surface receptors that the phages use for recognition, or developing nucleases that destroy the incoming phage DNA as the two major defense mechanisms. But the good news is that because this has been going on for 300 million years, there are there are so many phages on the planet, there's always another one around the corner to go after that same bacterium. And as these bacteria evolve, they will give up fitness as they make these changes to be able to adapt to a given phage in a given location. Now, over time, that fitness loss will be compensated for. But from the medical perspective, what happens is that we see selection of resistant bacteria, just like we do with phages, just like we do with antibiotics. But we can also take that same library of phages that were developed for that particular bacterial species and look for phages that are active against the resistant bacterium and come back in with a round 2 of phages. That's what we did with Tom's bacterium when we discovered that phages were that the bacteria were evolving that were resistant to the phages we were using. Now, in his case, one of the things we didn't know at the time was that the phages we were using from Texas A&M and also from the Navy were very similar. It's difficult to sometimes sequence phages because phages, to resist the endonucleases that the bacteria throw at them, methylate their genomes and do things that make it hard for us to use some of our commercial kits that we use to sequence phages. And the Navy, neither the Navy nor A&M had sequenced the phages. And when the phages were sequenced, it turned out to be they were really, I can say this is an Alabamian, as close together as West Virginia cousins. And so that as they began to evolve, they're really being selected by almost a single phage. We are working on approaches to develop cocktails more intelligently that would include phages of different classes. And in fact, the phage that was pulled to deal with Tom's resistant organism was one from a totally different phage class, and when put together with some of the original phages, suppress the organism very well. We're working on ways to both figure out how to do that up front more intelligently, and also to use phages in conjunction with antibiotics in ways that further limit the ability of bacteria to evolve. So I think it's going to be one of the things that we work out in the translational work that goes forward, using a lot of the principles we did with antiretroviral therapy.
Great. What are some of the ethical limitations in the development and perhaps the promotion of bacteriophage therapy?
Well, I guess there are several aspects of this. Steffanie already touched on one, and that is she was extremely fortunate to be sitting at as one of the Uber deans at UC San Diego with people who were willing to be manipulated into providing phage for her husband. And that's a very fortunate place to be. One of the things that we really want to be able to do is to operationalize this for much larger segments of the population, have it be mainstream, made available as a mainstream part of medicine in the US and indeed around the world, because phage is really not that expensive to produce. They're not that expensive to distribute. And once an infrastructure is in place, operationalizing this on a global scene, to me, is one of the things that is really an obligation we have, just like we did with antiretroviral therapy 25 years ago. As to concerns about the environment, there have been a lot of concerns raised that I think are probably overblown about using natural phages and worrying about having contamination of the environment. Because if you look at our own gastrointestinal tracts, we have about 10 to 100-fold more phages in the GI tract than we do bacteria. And when the organisms they are trying to attack are depleted, there's nothing for the phages to continue to expand with, and they themselves fade out. So we continuously have this ebbing and flowing of phage populations as we see as they encounter bacteria in nature. Now, there is work going into developing synthetic phages, and some of these are going to be ones that include Cas-CRISPR inserts and that have artificially concocted ligands for binding receptors on phages and on bacteria. We'll have to start thinking about how what impact they might have, the same way we do with 27th generation cephalosporins, in terms of whether they're going to be things that we should consider for specific conditions. Phages, by their nature, are highly specific. They for the most part, are more like using a laser than a cluster bomb and going after MDR infections. And so I think the ethical aspects that we dealt with, for example, with fattening up livestock, won't be as much of an issue with phages because of the nature of the phage bacterium interaction. A very nice article about phage ethics was put together by a visiting professor here at UCSD about a year and a half ago that I can send your way to get into it in more detail. But I think that we can solve most of these issues. There has been a lot of anxiety about, we had some anxiety in Britain about these being recombinant organisms, the phages that were used to treat Isabel. As Steffanie mentioned, Graham used a electroporation technique of his to remove the gene that allowed the phages to be lysogenic, the repressor gene. And the case we made to the regulatory agencies was that when you take a population of phages that are lysogenic, they will, just as the course of infection goes on, will have deletions that will result in loss of lysogenic capabilities, a couple of base pairs here and there. And the difference between the ones that Graham made, and the ones that happen in nature is that Graham took out a much larger segment and they couldn't revert back to wild type. So this was really just doing the same thing nature does, but more effectively. And we weren't adding DNA, we were subtracting it. So they didn't view those phages as genetically modified from the standpoint of their anxiety about Fruit Loops and things in the UK.
That's awesome. So given this, I mean, I have two questions and you can pick which one you want to take, but this is like blow you to the back of the theater kind of interesting stuff. So I'm glad you guys are out there talking about it, and thanks for coming in and talking about it today. But one of the questions is, since this is an experimental treatment, who pays? I mean, so when you do this sort of work, does your health insurance cover this as this goes on? Does Medicaid cover it? Or is it all cash business? And then the second question is, I think one of the things that Steph, you talked about at the beginning was, how does this have, what are the directions that relate directly to HIV? I mean, Chip, you've done sort of salvage therapy for a long time, and it sounds a little bit, I don't know if there's similar parallel logic or something, but because those are two questions that come to mind.
The cost issue so far has been a mix of a couple of different issues. Most of the phage hunts have been done by academic collaborators who have done it kind of out of a combination of scientific interest and compassion for individual people, and it's not sustainable. We have to find a way to, as Steffanie said, develop phage libraries that are widely available and that can be used without having to have academic laboratories do this each time a new case comes up. Preparing phages for clinical administration using good manufacturing practices has also been worked out pretty well, but it's not the kind of thing that academic labs like to do. Many of us are in academics because we don't like dealing with checklists, and we like to do things that are innovative and fun, and what the GMP is like a nightmare of having to go to mandatory trainings the rest of your life that all of our administrators make us take hour and hour over and over again. So we need to develop places that can efficiently develop GMP manufacturing capabilities for phages to be given clinically. The physician care has been generally provided in the context of normal clinical care. Infectious disease specialists, other places who are taking care of the patients or here are seeing the patients clinically, and they haven't been asking for infusion fees like the oncologists do for their chemotherapy. A lot of the work has been done for outpatients, and generally our outpatient pharmacies will pay for outpatient infusions. They don't really care what's in the infusion, so that a lot of times health insurance is paid for visiting nurses to provide support for the infusions. A lot of the patients do it themselves. It isn't yet mainstream, and so we haven't yet been, people haven't been being charged for the phage material, although some of the academic laboratories have come together with approaches to do that. In Baylor, they're doing it in a way that I think is basically cost neutral. Over the longer term, what we have to do is find a way to for want of a better term, commercialize this so that it's brought into the regular stream of healthcare expenses, and it's something that you don't have to worry about when you're in need of phages any more than you do about having them pull a bandage out of the hospital supply room. But we're not there. We're not there yet. And your other question was about the
Parallels to HIV.
Yeah, I guess, yeah, what we've learned from the HIV epidemic. I think many of us have take homes in this respect, but I think one of them is that, the HIV activists, created a parallel track when experimental treatments like, AZT initially were being studied. And that exists now as a result of some of the advocacy that they did. And so it's still possible to get compassionate use phage therapy, even though the clinical trials haven't been done. So that's something that we really owe a lot of HIV activists. Great.
Well, seriously, this is absolutely incredible. Thanks, Professor Strathdee and Schooley, for coming in and talking to us about this exciting just emerging area of science. This is fascinating. It's really wonderful. I want to thank you. And I also want to thank our partner, the UCLA AIDS Institute, for today's HIV Grand Rounds. I appreciate everyone joining. If there are any other questions, feel free to send them to us. The speakers will get them. Also, just reminding everybody that this will be posted on the CHIPS website. This talk has been recorded and will be posted on the CHIPS website sometime next week. So
thanks so much, Steve. And any of you, I mean, phage therapy is still experimental, but if any physicians have patients with multi-drug resistant bacterial infections that are not responding to treatment, feel free to reach out to us for a free consultation.
Awesome. Thanks, Steph. Thanks, Chip. We'll see you in a month.
Sounds good. See you later. Bye-bye.